Now today we will be starting with the lecture number 2 and we will discuss the immune system. The competency covered under this lecture is 10.3, which states describe the cellular and humoral components of the immune system. Learning objectives under this competency would be that student after studying this chapter should be able to enumerate the components of the immune system, and number 2, explain the role of cells and lymphoid tissue in immune system. Now let's talk about immune system. The immune system broadly is of two types: innate immune system and adaptive immune system. Innate is the first line of defense, and that we have discussed in lecture number 1. Now today we will be focusing on adaptive immune system, and in fact most of the discussion now on that will follow will be regarding the adaptive immune system. And also whatever generally we talk about immunity, immunology, development of immune response, histocompatibility, cellular, humoral, usually it concerns around adaptive immune system. So let us begin with the adaptive immune system. The major components of this system are various cells. Major among them are leukocytes, NK cells and APCs, also referred to as antigen presenting cells. We will be talking about them. The lymphoid tissues, which includes primary or the central lymphoid tissue, namely thymus and bone marrow, and secondary or peripheral lymphoid tissues including lymph nodes, spleen, GALT or the cutaneous associated lymphoid tissue, gut associated lymphoid tissues, etc. Peyer's patch is an example of gut associated lymphoid tissue. Then we have MHC molecules or the major histocompatibility molecules, concerned largely with the determination of self and non-self as well as development of immune response, and the various chemical mediators which bring about the immune response. These are immunoglobulins, complement proteins and cytokines. Now coming to lymphocytes, there are two main types of lymphocytes referred to as T lymphocytes and B lymphocytes, which are talked about very often and play a critical role in the adaptive immune system in the development of immunity of both cellular and humoral types. Now the picture on the right hand side shows the blood picture, in which the red arrow indicate a small lymphocytes, and if as you notice in this, these are usually 6 to 7 micron in size, and most of the lymphocyte is occupied by the darkly stained nucleus. The cytoplasm only is visible as a thin rim around the nucleus. Adjoining to that is a large lymphocyte, which is indicated by the black arrow, and you find this cell is relatively bigger and has got a relatively thicker margin of cytoplasm around the nucleus. Another cell is shown by the green arrow, which indicates a large granular lymphocytes, and you can clearly see the granule in the cytoplasm. This cell is quite large. Now this is nowadays called as NK cell, and we have already discussed about it, and we will further talk about NK cells when we come to cellular immune response. Now coming to these lymphocytes, so the peripheral blood has got two types of lymphocytes, the small lymphocytes and the large lymphocytes, and both T and B lymphocytes arise from the small lymphocytes. Now let us talk about these lymphocytes. Now B lymphocytes, its origin is in the bone marrow, and then it remains in the bone marrow itself, undergoes development and maturation to become a fully functional mature B cell or B lymphocytes. Now this cell is called navy B cell because it has not been exposed to any antigen. So a fully functional mature B cell which has not been exposed to the antigen as yet is called a navy B cell. Now going into the details of the structure, it is apparently under the light microscope, it is same as compared to the small lymphocyte that you see, because these other details are not visible. However, under large magnifying microscopes such as electron microscopes and other mechanisms, we can find the other details. Now each B cell on its surface contains large number of molecules. Unique among them is what is known as BCR, or the B cell receptor. Now this B cell receptor is present on each B cell. Now this B cell receptor acts as the antigen sensing and binding units. Through this receptor, each B cell comes in contact with the antigen and binds to it. And after the antigen has bound to the B cell through its receptor, B cell becomes active and undergoes proliferation to give rise to what are known as effector cells. Now this B cell receptor consists of two broad units. One is a antigen binding unit and another one is a signaling unit. The antigen binding unit is typically a membrane bound antibody, which can be either IgM or IgD variety. There are 5 types of antibody molecules present in our body, referred to as classes of antibodies, namely IgA, IgM, IgD, IgE, IgD. Now of these, IgD and IgM are present in the B cell receptors and act as antigen binding unit. They are attached to the membrane of the B cell. Through this receptor, as already said, the B cell binds antigen and becomes active and give rise to effector cells. Now this antigen binding unit or the antibody is highly specific and is able to bind a one specific unique antigen. So we can say that each B cell is specific for one particular antigen. And in our body it is estimated that there is a large number of antigen specific B cells, roughly about 10 to the power 7 to 10 to the power 9 types of B cells. And each of these cells is capable of binding to one specific antigen. So therefore it indicates that our body has got an enormously large population of B cell and it is almost able to take care of any antigen that it is likely to encounter in the body. So therefore this B cell receptor is a unique molecule responsible for antigen binding and activation of B cells. There are other molecules also, but this is unique, which is not present on other cells. Now I have already told you that the navy B cell, on binding to the antigen in the BCR, antigen binding unit, which is an antibody, undergoes activation and proliferation to become a activated B cell. And this activated B cell enlarges, called lymphoblast, undergoes proliferation to give rise to a clone of activated B cells. These activated B cells differentiate into two types of cells, the memory cell and the plasma cells. The plasma cell is the cell which produces the antibody. And remember, this antibody produced by the plasma cell is unique in the sense that its antigen binding specificity is identical to the original antigen which bound to the B cell and stimulated it, right? And the memory cell is the cell whose receptors on the surface have got the same antigen specificity as the original antigen which activated the original navy B lymphocytes. The plasma cell is a short-lived cell. It is rich in nucleus and other organelles, particularly endoplasmic reticulum and ribosomes, etc., because it synthesizes large number of antibodies which are gamma globulin in nature. So its essential function is that it has got enormous capacity to synthesize antibodies over a short period of time, and then it dies. However, latest development indicate that these cells do not die and they remain present even throughout the life. The memory cells, of course, remain dormant throughout the life, and by virtue of the antigen specific receptors on their surface, they remain sensitive or specific to the original antigen which bound to the B cell and activated it. So effectively we are saying that each B cell has got a unique antigen binding receptor through which it binds to one unique antigen, undergoes activation and proliferation, producing two types of effector cells. These effector cells are plasma cells and the memory cell. Memory cell remains dormant, however retains the antigen specificity of the original antigen, and the plasma cell produces antibodies which are unique in its specificity to the original antigen which activated the B cell, the navy B cell. So therefore the scheme is clear. B cells develop in the bone marrow, mature in the bone marrow, come out and circulate in the blood, bind antigen, get activated, undergo proliferation and produce effector cells, namely the plasma cell which produces large amount of antibodies, and the memory cells responsible for future memory. The picture in the sub-picture in the right hand side shows a plasma cell indicated by arrows. So that is the function. Now production of antibodies by B cell following the activation of the B cell by antigen binding is referred to as humoral response. So if I ask you what is humoral response, you should answer that production of antibodies following the binding of antigen to the B cell and its activation, leading to production of effector cells, the plasma cell producing antibody is called humoral response. So now you know the navy B cell, the activated B cell, the effector cells and the response of the effector cells. So that much is about the B cell. Now coming to T lymphocytes, these also are arising from the small lymphocytes. Their origin is in the bone marrow. However, a set of developing thymocytes in the bone marrow migrate to the thymus and they develop in the thymus and mature in the thymus to become T lymphocytes. If thymus is not there, it is seen that the T cells are almost absent in the blood. There is a clinical condition called Dye-George syndrome in which there is either complete agenesis of thymus or it is atrophic or very small sized in development and does not perform its adequate functions. Now in such systems the T cells are totally absent in the blood or are present in very small number. So that indicates that the thymus is essential for the development of T lymphocytes. So they are small lymphocytes, and one set of developing small lymphocytes remain in the bone marrow, mature there itself to become B lymphocytes. The other set moves to the thymus, develops in the thymus, mature in the thymus, becomes T cell. Now, like the B cell, the fully functional mature T cell without any exposure to antigen is called a navy T cell. And comparable to the B cell, it has also got many surface molecules, and the unique among them is called the T cell receptor. And this receptor also has got a central binding unit or the antigen recognition unit associated with the signal transduction unit indicated here as CD3. So the composition of the T cell receptor is also comparable. However, it is to be remembered that the antigen binding unit is not an antibody unit here, but it is comparable to them. We will see in the next slide. So, like the B cell, the T cell, after coming in contact with the antigen, the antigen is bound by the T cell receptor, and on antigen binding, T cell signal is transducted through the signaling units, and it undergoes proliferation and production of effector cells. The effector cells produced by the T cells are cytotoxic T lymphocytes and memory cells. The memory cells, like B cell, remain dormant, carry the same receptor with the same antigen specificity which originally activated the B cell, and the cytotoxic T cells are the effector cells which are developed from cytotoxic T cell precursors. This slide just shows the comparison of TCR and BCR. They are not identical, but they are similar. The pattern of design is that each has got an antigen binding unit and a signal transduction unit, and in fact, as if you see the details, the signal transduction unit is a multi-molecular unit. It's not a single molecule. The antigen binding, of course, is a single molecule. In case of B cell receptor, the antigen binding unit is a membrane bound antibody, either IgM or IgD. But in case of T cell receptor, it is not antibody, but it is similar to antibody molecule, consisting of two polypeptide chains alpha and beta, and having a structure comparable to antibody molecule with a antigen binding sites on them. Now let us talk about T cell subsets, or subsets refer to the different type of T cells present, and it is very important to know them because they have very important and differentiating roles in the development of immune responses. They are identified on the basis of molecules present on the surface, which are called as CD markers or clusters of differentiation. Often there are more than one molecules which are used to identify a particular cell. So like that, several molecules were identified, and a group of molecules which identified a particular cell or a molecule or a stage of developing cell was clustered together as cluster of differentiation. That is how this came as CD markers. Also, during the course of development of immunology and identification of these cells, it was also observed that the same molecule was often identified by different names because of use of monoclonal antibodies by individual labs. So there was a lot of confusion. So first international workshop on HLA, that is human leukocyte antigen, in 1985 studies all such markers and ultimately grouped them and classified them to remove that ambiguity. And to date, more than 350 such markers are known. We will be talking about some of the critical markers on the basis of which major T cell subsets are identified. Now there are three main T cells on the basis of CD markers. One is T helper cell, another one is T cytotoxic cell, and the third one is T regulatory cells. T regulatory cell was earlier called as T suppressor cell. Now coming to T helper cell, it carries a marker called CD4 on its surface. So all T helper cells are CD4 positive cells. They are simply at times called as CD4 positive cells. They belong to T lymphocytes and carry CD4. They have all the other properties of T lymphocytes. And their role is unique because they help in activation of both B cell to produce immune response of the humoral variety, and they also help in the activation of TC cell to produce cellular immune response. So T helper cell or TH cells are CD4 positive cells required for activation of B cell and TC cell to produce both humoral and cellular immune response. T cytotoxic cells, also called TC cell, they are CD8 positive lymphocytes of the T lymphocyte variety, and they produce on activation a effector TC cell, that is CTL, cytotoxic T lymphocytes. So TC cell on activation becomes CTL, that is cytotoxic T lymphocyte, and cytotoxic T lymphocytes are responsible for cellular immune response. So TC cell is directly responsible for the activation of cellular immune response. Now you know that TH cell also needs to activate TC cell and therefore to produce cellular immune response. So TH cell is critical for both humoral response as well as cellular immune response. T regulatory cell is also CD4 positive cell, but it carries another positive marker on its molecule, that is CD25. It is basically a regulatory cell which suppresses the immune response, particularly the inflammatory immune response. So therefore this is also a subset of T cell. We have already talked about it. So these are the three major subsets of T cells. To repeat again, TH cell, which are CD4 positive, needed for both humoral immune response and cellular response. TC cell, CD8 positive cells, needed for directly responsible for cellular immune response, and T regulatory cell, which are both CD4 positive and CD25 positive and are responsible for suppression or regulation of immune response, especially we need to talk further about the various types of TH cells themselves. Means there are different types of T helper cells, and as you shall see, this is very important to understand the basic mechanism of development of immune response. There are several other types of TH cells, but here we will talk about three major types. All are CD4 positive cells because they are all TH cells, and they are TH1 cell, TH2 cell and T regulatory cell. T regulatory cell we have already talked little bit, we'll elaborate little more here. Now remember, these cells have got clear-cut functions in the immune response. They all belong to TH cells, they all are CD4 positive, and they are identified on the basis of their functional properties, which is the production of cytokines by them. A set of cytokine produced by those cells, these cells are called as cytokine profile. And these cells, during the process of interaction and binding with the antigen under different conditions, are differentiated into different TH cell subsets. For example, a TH cell binding to an intracellular organism or reacting to an organism which is intracellular in nature often leads to differentiation of the TH cell to TH1 cell. And it is the environment of the interaction between the TH cell and the antigen and the present cytokines in that environment which determines whether the cell will differentiate into TH1 and TH2. And they are identified on the basis of the cytokines they are produced after their differentiation. So TH1 cells produce a profile which is referred to as TH1 cytokine profile, and it consists of interleukin 2, TNF alpha and interferon gamma. So these are TH1 cells, and their very important role is that they activate TC cells, which becomes then cytotoxic T lymphocyte, that is the effector TC cell, and therefore produces cellular immune response. Similarly, under different sets of condition of interaction with an antigen and the environmental profile of cytokines present during that interaction, the TH cells might also differentiate into what is known as TH2 cell, and they are identified by their cytokine profile called as TH2 cytokine profile, and typically they produce IL-4, 5, 6, 10, etc. And TH2 typically activate B cell into their effector cells, that is plasma cells and memory cells, to produce humoral immune response. So therefore, T cells are first divided into two types: T helper cell, T cytotoxic cells. TH cells are further divided into TH1 cell and TH2 cell. And TH1 cell activate TC cell to lead to cellular immune response, and TH2 cell activate B cell to lead to humoral immune response. Now this is important basic fundamentals to understand. They may be structurally same under usual conditions of identification. However, their secretion of cytokines profile is different, and on the basis of these cytokines they are identified. We have already talked about T regulatory cells. They are CD4 positive, CD25 positive cells. They suppress immune response, and their cytokine profile is IL-10 and transforming growth factor beta. Transforming growth factor beta. These cells suppress APC activity, antigen presenting cells activity, and they also regulate the inflammatory response. Inflammatory response is largely a part of cellular immune response, so they regulate in a way the cellular immune response. So that is how you have the different sets of T cells and then different subsets of TH cells. If you read some books, you might find some more subsets of TH cells, namely TH3, that is TH17 cells, and TH4 or the TH follicular cells. However, at this stage and at your level, those details are not required.